Hi, this is Ms. Slitton, and this is the final review for Honors Bio, and this is Unit 8. And Unit 8, sweetheart, she needs to be separate from you. Oh, okay. Unit 8 was a hyperdoc about stress, right? And that is kind of standalone. I don't know that I asked a specific question on stress, so you can call that the end of that on the stress. And the other part of Unit 8 was the biotechnology part. And I did review that, the big aspects of biotechnology in, I think the Unit 7 review or 6 review. So I would revisit that because I went over that. And then I'm going to specifically go over the Amgen lab with you today. And then once we finish reviewing the Amgen lab, then you can just ask me general questions you might have. What's so funny? Really annoying children are in this room right now? Children who are annoying?
You're having a great day.
Focus. Bear down for you. All right. Okay, yeah. All right, so let's talk through the parts of the Amgen lab to make sure we have that. The first part is that we need to understand antibiotic resistance, that when bacteria have a gene, right, that gets transcribed and translated that protects them against an antibiotic, then that antibiotic can't kill them. But they have to have the DNA to code for the proteins that will basically disable that antibiotic. As long as they have that, then they live. There are two plasmids that we're dealing with in our lab. The first plasmid that we dealt with was pecan, and pecan had kanamycin resistance. Has kanamycin resistance. And that trait can be expressed, but there's another gene in pecan that's not expressed. That's the RFP gene. Why isn't that RFP gene expressed? Why don't you, if that plasmid was in bacteria, if you gave them pecan, why wouldn't they glow red?
They don't have a switch.
They don't have a switch to do what?
To
turn on the gene, right? To turn on the gene, exactly. Then the second plasmid that we dealt with, okay, was Piara. Now, Piara has a switch, right, but there's no specific gene right next to that switch to turn on using the switch, right? But it also has an antibiotic resistance. It has ampicillin resistance. So if you put it on a plate that has ampicillin, it will survive. Now, why would they ever use antibiotic resistance in biotechnology? Because that doesn't sound smart to make antibiotic-resistant bacteria. Why would we use that? You know why? It's a way to know, it's an easy way. It inhibits the growth of every single bacterium that didn't get a plasmid, right? Because the only way you can survive on that plate is if you have a plasmid that contains antibiotic resistance. So all the other bacteria are killed off, okay? Except for what kind of plate? Where can all bacteria live? On the LB plate, because it's just plain up food. And that plate, why did we use that plate in our lab at the end? It was a control, but what were we checking to see?
If
they're alive, right? We just want to even know if we have living bacteria, okay? And that plate will verify that both your P plus and your P minus bacteria were alive and they could grow on that plate. All right, so I was just kind of looking forward to that. So what we were doing in our lab was we were trying to cut out of the pecan, trying to cut out this RFP gene. What do we use to cut DNA? Restriction enzymes. Where do they cut? At recognition sequences, specific series of bases, right? And once we cut them, right, what does it leave when you cut it? Sticky ends. What's the beauty of a sticky end? Another sticky end that starts with a C, complementary. If it's complementary, then you can line them up together. Now, when you line them up together, what do you need to do to make sure they stay stuck together? Ligase, good, the chemical tape. So what we were trying to do is cut out this RFP gene and insert it over here next to a switch, and that switch was the ARA-C gene. And so as long as bacteria are in the presence of arabinose, then they will turn on the neighboring gene as well. Not only will they turn on the arabinose gene, but they'll turn on the neighboring gene. And that's what we are attempting to make. And if we made this, it would actually be called PRR, because it's recombinant PRR. It has a new gene in it. Okay, look to your person next to you and say, do you understand everything she said so far? Do you have any clarifying questions, anything she said so far? Okay, now ask me. Yes?
So pecan doesn't have the switch, so
it doesn't have it has switch for pecan. You can't see it, but it is actually there. But it didn't have the switch for the RFP gene.
What's the switch for the RFP?
Yeah, the ARA-C gene. When you have arabinose present, it'll turn that gene on and turn on the neighboring gene. Yeah. Okay, does that make sense to you? Okay. All right. Was there any question your bio buddy didn't answer for you? Anything you want to ask me? Only got one? Okay. I don't know what's taking so long. Yeah, go ask again, but try not to be like buggy. Okay, I know you do it right. All right. Okay, so we were using 2 different restriction enzymes to cut our plasmids. Why? Remember why? Yeah, we didn't want it to invert. We wanted that RFP to go in one way. We wanted it to go in RFP, not PFR. Yeah. Does that make sense? We didn't want it to go in PFR. We wanted it to go in RFP. All right. So here you can see a picture, and you can see that when we cut the 2 plasmids, we would get 2 fragments from each of them if we cut them in the right place. And they were genetically engineered with those restriction enzymes, those recognition sites in that spot. Did she give you a Scantron, or do you need one? Okay. So while I'm getting her a Scantron, talk about this page, oldest bio buddy, go.
Okay, so. Oh, okay.
Will you do me a favor? My next room is just going to ask if I can have a solution that she just gave that to us. Thank you. All right. So you get 2 fragments here. Now these plasmids were engineered, right, to have those recognition spots in those places, those recognition sites. So chances are they were made correctly. But if you did in fact cut your 2 plasmids, you should get a large and a small fragment. Now, how would we know? Could we ever look in the test tubes to see those fragments?
No.
So how do we verify that we've actually cut our DNA?
Gel electrophoresis.
Okay, because whole plasmid will travel differently than cut plasmid, right? When we do gel electrophoresis. And then looking forward, seeing what we remember, how would we know we cut them in the right spot where we got the right size fragments? What would we do? We would compare it to the lambda BC marker. We compare it to the marker DNA, right? 10 known size fragments. And how would we know we ligated our plasmids and got new combinations, or even reformed the old combinations when we mixed them all together?
If the red was shown.
No, not if they're red. How could we tell in the gel that we ligated? Do you remember what would be in the lig row? They'd be whole plasmids, right? And we'd have a bunch of them, so it'd be like a skid mark right there in the lid. Does that make sense? Okay, so I'm just kind of thinking forward on how you would be able to tell the difference. So, thank you so much. Okay, thank you. When you put the restrictions enzymes in there, we let them sit for a couple hours so they could do the cutting, right? And then we pulled them out and I stuck them in the freezer. Okay, so that was the digestion day. The following day was going to be our ligation day. But before we even added any chemical tape to this, we had to do something else first. What was the something else first we had to do? Denature. Inactivation. What were we inactivating? Restriction enzymes, because we couldn't pull the scissors out of those tubes. So instead, we took the tubes and put them in hot water to denature the restriction enzymes. Okay, and so once we put our A plus and our K plus, and I think we had room, we put all our tubes in there just to keep it consistent, right? Then we could do the taping. We put some ligase in the bottom of a tube, the chemical tape. You added from your A plus and your K plus. Put a little buffer in there to maintain the pH. and then you let it sit overnight. And at that point, you had 4 different fragments that could combine in any which way. Could you have reformed your original plasmids?
Yes.
Yes. Could you have formed new ones?
Yes.
Yes, hopefully we did. Because we wanted the RFP fragment from pecan to hook up with the P-aura and the ampicillin plasmid, right? And so that we could form that one that would allow us to express that gene. So that was day 2, and that was the ligation day. And we call that chimera day because we could make any new combination. And this was the combination that we were after. So if we were successful, and if we put this plasmid into an E. coli, okay, that particular plasmid into an E. coli on transformation day, then it would, because if a rhabinose was in where the bacteria was living, if we had a rhabinose sugar would turn on the P-aura gene, which would then turn on the RFP gene, which would then start transcribing and translating and making that red fluorescent protein. Okay, yes?
The P-aura gene already have a rhabinose, correct?
Yes. Okay, turn toward your bio buddy and see, was there anything there that you were struggling with or you didn't understand that I just explained? Okay, clarifying questions you want to ask me? Yes. Wait, say it out loud so everybody else can hear it.
So that the rhabinose sugar would turn on the P-aura gene, then what does it
Turn on the RSC gene,
Oh.
which would then turn on the RFP gene. Okay. Any other clarifying questions on that? Okay, so let's move forward to the next day. So we had digestion day. We had practice day, right, with dyes. I'm not really counting that. But as far as the lab goes, we had digestion, cutting day. We had taping day, inactivation and taping day. Then the 3rd day of the actual lab, we did gel electrophoresis. So we wanted to verify, did we cut the plasmids? Well, we'll compare the way the gel turns out with cut and uncut, and they should look markedly different. We could predict what it would look like to know if we had cut it. We would compare our cut DNA with the marker to know if we cut them in the right places. And then the 3rd one is, did we ligate to form new plasmids? We would look in that lig column and see if we had a bunch of skid marks in there of all the different whole plasmids we had made. Okay, turn toward your bio buddy, youngest bio buddy. Put this in your own words. See if you can understand that. Go ahead, try to put in your own words, youngest bio buddy. Okay, so this part was just kind of the explanation of how to do it and how to load it. But in essence, what we did is we pulled samples of DNA out of our A+, A-, K+, K-, lig2, right? Those are 5 sample DNA, experimental DNA, and we also had the marker tube. That was our 6th tube. And for us, we had to also load in that loading dye that would make it glow, right, in the mini one. And then we put those in the gel, loaded them in, and we turned on that current. And then we were looking to see how it would turn out. And, oh, no? Where's that little embedded picture? Here, that's what I want. Okay, so if we take a look at this picture, take a moment just to absorb that. Just look at the K- and the K+ rows. Look at the K- and the K+ rows. Do they look different from each other? Yeah. So the K+ looks, these are the 2 fragments of the K+. Here, right here, the reason why we see skid marks, even though it's only one kind of plasmid, is because they travel in different, ways. Some are like this, some are open, some are nicked, some are multimer, some are supercoiled, right? So they will travel, but they're all the same size plasmid, not cut. If we look at A-, A+, can we see a difference? And we had a hard time visualizing these 2. That's not your fault. We just had a hard time visualizing that loading dye, just kind of bleached out too much. But we could still compare, like if you just looked at K-, K+, it looked different, and A-, and A+, it looked different. Okay, and then the lig tube, these were just a bunch of large plasmids, and they didn't travel very far, and you have various sizes. And then your marker was here across the top, and then we could line up where we would predict each of the fragments to be compared to the marker. Yes?
What does the DL on the top stand for?
DNA ladder. Yeah, that's your DNA ladder of known size pieces of DNA. Okay, ask your neighbor, are you okay with this picture? Do you understand it? Clarifying questions? Kurt was dancing the whole way to the car. I don't know who she is, but she's super cute. Okay, yes.
Is the DNA ladder the marker?
Yes, the DNA ladder is the marker.
And what is the lig called?
The lig could be any combination of the 4 fragments together, right? You could reform your original plasmids, any combinations you could have. Okay? Other questions on that? All right. So then that brought us to the last 2 days of the lab. And what we are trying to do here is take all the different things out of our lig tube, all the different combinations we have, and transform bacteria with those different combinations, knowing we could get a lot of varieties of E. coli there, but we were searching for one, the one that had the PRR plasmid that we wanted. So you received competent cells that you kept real cold, and then to one of them you added plasmid, and the other one you didn't. And so the one that got the plasmid is the only one that should be transformed. The P- got no plasmid, no special powers. The only plate it should be able to grow on is a luria broth plate. Okay? And the one that had P+, you should have options here. And after you iced them, you did a heat shock. What was the significance of heat shock?
Get the plasmid inside.
Yeah, get the plasmid. Helps the pressure differential will help push that plasmid inside. And this heat shock is different from inactivation, right? Inactivation was really inactive. It was really hot. It was a 70-degree water bath. You're trying to denature the restriction enzyme. In heat shock, it's just a 42-degree water bath, and it's just trying to go from cold to hot back to cold to kind of force it in there. Then what did we do to them after we heat shocked them? Do you remember what we did to them? We gave them some. We put them back on the ice for a couple minutes, and then we gave them some what? Luria broth. Yeah, you fed them both. You fed them, and then you put them in a nice warm water bath inside the incubator so they could do a little asexual reproduction, a little binary fission, and make another whole copy of themselves with all that genetic information in it. So now you should have twice as many bacteria as a result of that. And then all you had to do is, and you mixed it, right? Most of you. And then what you had to do is just plate it out, okay? So P- only got to go on the LB plate and the LB amp plate. And that would just verify that they could not naturally grow on a plate that has ampicillin. They could only grow on that plate if they were in fact resistant to that antibiotic, right? Yes? And then you put your P+ on the LB plate, you put it on the ampicillin plate, but additionally you put it on a plate with arabinose. And that plate with arabinose, that's the only plate you should be able to see red colonies. You could have had the possibility in the P+ on the LB amp plate, you could have had the possibility of the P+ just on the LB plate. But we don't have a way to turn it on unless we're on the arabinose plate. And the antibiotic, it was just there to eliminate any bacteria that did not have the ampicillin resistance so that we could just focus on can you glow or not, right? Because you could have received ampicillin resistance and not received the RFP gene. The original Piara, right, the original Piara was resistant to ampicillin. It just couldn't glow red. Thank you. Oh, that's yours. Okay, yes?
Why does it need arabinose if it has the gene?
Okay, so let's go back to our original pecan, right? Do you remember when we learned, and we reviewed this in a review, I think two days ago, RNA polymerase, what, binds to the promoter.
Oh, okay.
Remember, repressors can get on the operator and interfere with that. So by having arabinose present, think of it acting like an inducer to get that to move forward. Okay, so then we plated it on those different ones, and then we predicted results, and we predicted that you would get massive growth on the LB plate, whether you're a P- or P+, because there was nothing to stop bacteria from growing. So we got growth on both halves. And then when we looked at a plate that had ampicillin on it, we would only expect growth on the P+ side, not on the P- side, because the bacteria on the P- side didn't have any ability to create any protein that makes them resistant to ampicillin. Only the P+ ones did. And then on the arabinose plate, that's the only one we would expect to have red colonies. Okay, ask your bio buddy if you understand the procedures of the lab and what we did there in the lab. Clarifying questions you have about the lab. Okay, now I'm just open to general questions you just might have about other things. If I've covered it in another review, I'm going to encourage you to watch that other review. But if there's something I haven't discussed yet that you want to go over, something flying around in the room? Because I keep seeing bugs. Yeah. Go away, bugs. Okay. Questions you might have? Yes.
We're studying the pig lab for the time.
You know what? I didn't put something specifically about the pig on there. See, cool. What else? Yes.
It's not a question about this, but is the final worth 20%?
I don't do it like that.
Oh, okay.
Do you remember what the final is worth, my friend?
60 out of 160.
Yeah, you can get 160 points out of
Oh,
150 points. Yeah, I make things, I weight things based on, yes? Why weren't they in the review today? I weight things based on how many points I give it. And that way just points go into the points pot, and if you get enough of those points, you get an A, and if you get some more points, maybe you get a B, whatever, yeah.
Will the environmental concerns part of Chapter 6 be on the final report?
Yes.
Was it in one of the review sessions?
No, it's because we just did it, so I figure you would know it.
Okay.
So on your environmental concerns, we looked at the ways that man kind of takes from the environment. So we looked at things like land use, water, food, right, and overexploitation of that. And then we looked at things that cause extinctions. Do you remember what causes extinction? What's the #1 cause of extinction involved in habitat loss? Like an 85%, right? Then who was number 2?
Exotic species.
Exotic species, number 2. Who was number 3? Pollution. How much was exotic species? Like 50%. And pollution was like around 24%, right? And then who comes next? Overexploitation, and then disease. Okay? And then we talked about, also in the beginning we talked about renewable and non-renewable resources, right? And how we should maybe focus potentially on using more of the renewable resources, right? And then we talked about the importance of biodiversity, and we talked about what were the 2 big categories of importance? Direct and indirect. Direct, the word you want to remember is MAC, because that's the acronym that will help you. The direct value of biodiversity. What was the M? Medicinal, yeah, medicines. What was the A? Agricultural, and then C was consumptive. Those are the direct values, the ones you can usually see. You know, there's like, I have this, I get money, right? I buy medicine, I eat food, I use rubber from trees, right? And then the indirect value is we were looking at things like what? Do you remember some of the indirect values of biodiversity? Waste disposal, phytoremediation. We looked at, what else?
Tourism.
What? Ecotourism. He knows everything he's been studying. Yeah, so go and compare your direct and indirect, right? And then we talked about remediation, too, right? We talked about keeping the environment that we have now, trying to maintain it, and then fixing the ones we need to fix as quickly as possible, the restoration part. One of the other things we talked about besides land use and water use was pollution. Remember we looked at all the different ways we pollute? What else? Good question. That it? Okay, and then remember my stream of consciousness words, right? So I would know what all those words mean, just bare minimum, for sure. Okay, what else? Yes?
Are there going to be problems like how...
Like genetics problems?
Yeah.
Yes. There will be genetics problems. Yeah. You remember how to do your Punnett squares. Do you want me to do one?
Sure.
Yes.
Okay. All right. So, what did Mendel work with a lot? Peas. He worked with peas. Cross two heterozygote pea plants for me and give me geno and phenotype ratios. Go. Okay, not out loud. Do it independent. Or in your head. Did I record this? Yeah, I did. Okay, I'm just making sure. Are you crossing it?
Yeah.
That is interesting. Okay, so how would I write heterozygous pea plants for height? Big T, little t with big T, little t, right? So when I do my Punnett square, whoops, sorry, I got a little spastic. We good on that? With those letters, what do those letters represent?
Dominance.
Okay, what else? More. Genotype. What would be the phenotype of this individual be? Tall would be the, so equals pheno. Okay, and what would this one be? Tall. So that was ratio 3:1 for phenotype. What's the genotype ratio? Yeah?
Oh, yeah. Okay.
See how I did that?
Yeah.
1 big T, 2 big T, little t, 1 little t, little t. Got it? That make sense? Okay. Cross for me. Oh, I didn't mean to do that. Cross for me. A woman who is this is a sex-linked trait. A woman who's a carrier for colorblind. Cross with a normal male. And what do you think is going to happen to their offspring? Do that one. I have a CB woman, and she's having sex with a normal male. And she's making children. How would you do that? Yes.
Are there going to be like diagrams of the body systems?
I don't remember if I put body system diagrams on them. Things related to them, maybe, or asking about them. Did you come to the review when I went over this? You want me to do this with you?
Yes.
Okay. So a color, oh, I said it was a carrier female, right?
Yeah.
That says colorblind, but I meant carrier. So female, XX, and so she's, what letter do you want to use? A? Okay. So she's a carrier. So is she colorblind? No. If she was colorblind, how would we write her? She's a carrier when she's like this. Okay, yeah, they're always A's. Give me a different, more descriptive. Little a, she would be colorblind. Remember, the sex-linked diseases I gave you are all recessive, right? And if it's a normal male, what does he look like? Yep. Like that? Okay, so if I were going to do a Punnett square, and I had the female was this, she's just a carrier, and the male is this, then this would be, oh, a mess. What would that be? What would that be? A normal female. Yeah, and this would be a phenotype-wise, she'd be normal. All right, what about this one? Yep, this female, she'd still be a female, but she'd be a carrier. Okay, what about this one? Okay, male, norm. And what about this one? Yep. Colorblind male. Okay, how do you feel about that?
Can you do the one with the blood types?
Sure. Okay, so I would like you to cross an AB with an O and give me their genotype and phenotype of their offspring. AB, person with an O, give me genotypes and phenotypes of their offspring. Geno and pheno. An AB crossed with an O.
So it'll be IA, and then the big I. Is it gonna be big right? It's two I.
Okay, so tell me. Come back to me. How would I write AB with the alleles? Be more specific. Big IA crossed with what? Little I, right? So if I did a Punnett square, yes?
So you put an X, you put like IA and then like X be like what? Okay.
That's a little tricky. That was a little tricky. Okay, so now, go ahead.
I think the bottom to the right is wrong.
You put I.
Yeah.
It's gonna be IB.
It would be IBI.
Yeah.
The right hand corner. The bottom right hand corner.
Like the
Yeah.
double I, like
Miss Linton, you are just lagging. Okay, so this genotype right here, big IA, little I, what would be the phenotype? A. Next one, big IB, little I, what would be the phenotype? B. And what's this one?
Same as the first.
Same as, and then this one. Yes, sorry about that. I wasn't even thinking.
What would be the phenotypic ratio and the genotypic ratio?
Okay, so let's do that. What is how many different genotypes do we have? Two. So it'd be 1 to 1. And how many different phenotypes do we have? Two. What would it be? 1 to 1. And then you would tell me what each of the ones represented. You know, throw in an X and something else in there, you know. What else?
And the I is because the O, that's what the O represents.
Little I is O, yeah. Tell me this. Let's do this. Okay, how are these different? What could these possibly be representing?
Well, the top.
Oh.
Yeah, gamete versus.
What would the top one be?
Gamete.
Yeah, this would be a gamete, right? What would this be?
Genotype.
A genotype,
Yeah.
right? What's the difference? A genotype, you have 2 alleles for every single trait, right? A gamete, you only have 1, right? Because you've done what process? Meiosis. When did you separate homologous pairs?
Meiosis. Oh,
Anaphase 1 of meiosis 1. When did you separate the sister chromatids? Anaphase 2 of meiosis 2. Good. Anything else?
I have a question.
Yes.
A monohybrid cross, is it strictly?
A hybrid, that would be like if you crossed, it's one trait. A dihybrid would be if you crossed like this crossed with whatever.
So they have to be heterozygous for it to be.
Well, you know what, that's a general term. Usually they're heterozygotes, but they're just meaning one trait cross, two trait cross. But if you hear dihybrid, they're specifically big Q, big R, little r. Two hybrid for each trait. Heterozygotes for each trait. Okay, what else? Feel good?
Yeah.
All right, I'm proud of you. Study that stream of consciousness that I have. Sure you're good with that. Have a piece of toast. Don't stay up too late. Love you very much.